Good afternoon, students. It is so good to have you in General Chemistry 1 this afternoon. I am so excited. I am so thrilled. I am so honored, and I am so privileged to have you as my students this semester. As we will begin, I want to remind everyone that you are not alone. This is an academic community. Remember to get help when needed. Reach out to university services if needed. Never give up. Keep trying. We are here to help you be ethical, honest, intelligent, and successful scientists. However, at the end of the day, you must be responsible, ethical, and hardworking. As we begin, I always like to reflect on the work of Werner Heisenberg. He was during his postdoctoral assistantship with Niels Bohr, um, Heisenberg formulated his famous uncertainty principle. At the age of 25, he became the chair in theoretical physics at the University of Leipzig. At 32, he was one of the youngest scientists to receive the Nobel Prize. He lived from 1901 to 1976. So this has been encourage and inspire you, hopefully, that it's possible, no matter how young you are. So, let's proceed. We're going to go straight to, I'd like to begin the lecture, discussing the significance of DNA. So I'm just going to go ahead and talk about it. So, for DNA, we know that in this class I tend to ask you um what functional groups do you see, which atoms do you see, and why is DNA important? So I'm going to give you some time and I'm going to give you the opportunity to go through the structure and just think about those answers. Which atoms do you see? What functional groups do you see? And why is this molecule, this polymer, why is it important? Okay, so let's begin. So right now we see that we have nitrogen. We see that we have nitrogen. We have nitrogen. Let me see. There we go. We have nitrogen. Nitrogen. We also have, wait a second, having a few technical difficulties. So let me see if I can adjust something quickly before I continue with the lecture for today. Adjust something quickly. Okay, let's proceed. So we have nitrogen, we have phosphorus, nitrogen, we have phosphorus. We also have hydrogen, we also have carbon, and we also have oxygen. Yeah, so that covers basically all of them we have here. Nitrogen, hydrogen, oxygen, phosphorus, and carbon. So those are the answers we see. The functional groups that we see, we have the amine. Is the amine there? Is the amine there? Right there. There we go. We have the amine there. See? There we go. And then we also have the phosphate there. And then we have other functional groups, but I'm not going into all that. We have hydroxyls there. And that's as far as I want to go with you today. So why is DNA important? DNA is important for several reasons. One, plays a role in the replication of cells. It plays a role in maintenance and integrity of cellular function. DNA is important because mutations in DNA can cause diseases, can lead to downstream effects such as diseases. DNA is very, very important. It's like the information, the set of instructions, the blueprint for a specific cell. Not every gene is expressed in every cell, and that's a discussion for a different day. But gene expression is very dependent, heavily dependent on the genes, and genes are made up of DNA. So, very important biopolymer. So let me erase, and we'll proceed forward. Let's see. So yeah, DNA is very important. It has contributed to a lot of discovery. You could go into all of the discussion as to whether it being semi-conservative in terms of its replication, all those things. But that's not for this class today. So let's talk about the goals and objectives of this class. I'd like to remind everyone of this because it's important for us to keep focus. The goal of the class is to teach the chemistry content in a way that is relevant to the Bahamian student and digestible for their understanding. The sequence is as follows. Understand the fundamental concept A, practice problems relevant to understanding concept A, learn more nuanced details about each concept, and practice more complex problems that integrate the details and the fundamental understanding. So today, we're actually going to go straight into our discussion for this lecture. This is just previous ideas that we discussed earlier. There we go. So I've already gone through the discussion in which we drew resonance structures, drew different Lewis dot structures. One of the things I want to do today is I want to emphasize some key ideas associated with resonance and resonance structures. So the resonance hybrid is one of the best qualitative descriptors of the molecule. And it gives us an idea. It points to the fact that electrons are not in one position all the time. It gives us it's a better description, a better treatment, of a better it leans closer and closer to a quantum mechanical treatment of molecular structure. It gives us an idea. It points to the fact that electrons are delocalized. It's a good structure. Not perfect, but it's a good description. And one of the things I want to emphasize in this lecture today is I want us to look at the resonance structures of nitrate. And so look at the resonance structures of nitrate. So let me, right here, nitrate. I want to look at the resonance structures of, yeah, I'll be focused on today in terms of resonance structures, and then we'll proceed forward into molecular geometry, and we'll continue to discuss some more ideas in terms of molecular orbital theory and valence bond theory. So let me erase, and now let's proceed. There we go. So let's talk about nitrate. So nitrate, NO3 minus. So we know when we look at nitrate, nitrogen is in group 5. So that's 5 valence electron. Oxygen is in group 6. So that's 6 valence electrons. So when we add those up, 18 plus 5 is 23. And then we consider the 1 that comes from the minus charge, so plus 1. And that gives us 24. That's that. So let's just so we have that in mind. So we're counting for 24 electrons. So let's come on this side right there. let's draw out we think the structure would look like. So we consider all of our electrons, we take our atom, we place it there, put single bonds between each atom. So NO, NO, let's see, let me just try just a little bit better. Actually, I can do a little bit better than that. So let me erase. Let's erase that. There we go. And let's erase that. And let's do this. And like this. There we go. Okay, so that's that. We draw single bonds. So 24 minus 2, 4, 6. 24 minus 6 is 18. So we put 1, 2, 3, 4, 5. 2, 3, 4, 5. 2. And then okay. So 6 dots or anything, and that accounts for all of the things. However, accounts for all of the electrons. However, the issue that we have here is that this has a minus charge, rest formal charge of -1. This has a formal charge of -1. This has a formal charge of -1, and this has a formal charge of +2. Though this is a resonance structure, it's not the best resonance structure that we can draw. Because one of the aims for drawing resonance structures is you want to make sure you follow the octet rule. That's a big chief idea. And you also want to make sure that you minimize charge, minimize formal charge. So that's one, but let's look at another one. So that's one we can draw. But let's look at another one. So let's look at another one. Okay. So this is a better one right here. See right here. This is a good one right here. It has an overall charge of -1, and we've minimized formal charge as best as possible. Everything obeys the octet rule. So we've ticked all of the boxes. So this is a good structure right here. You notice that we were able to draw two structures. You can draw different structures in which you have. So say for example, just keep that structure in your mind. We can draw a structure where we have the double bond here. So that. Um, and then we have a single bond here. And we have same nitrogen cells, the same form of charge with the double bonds change position. So as you can see, that's resonance structure, that's resonance structure, this is resonance structure. There's several you can draw. However, the best structure is one that minimizes formal charge and obeys the octet rule. And you want to make sure you hit all of those criteria when you're drawing resonance structures. Now we can draw another one, which we have the double bond on the other oxygen. So there, that, that, there. Actually, let me not rush this. Let me take my time and draw this clearly. So there you have. There we go. There we go. There we go. There we go. That. There we go. So that's another resonance structure. So we just drew four structures for the same molecule, for the same polyatomic ion. We drew four different Lewis dot structures. Different in the sense that the positions of the electrons are the position of the electrons between each atom, it's a slightly different configuration or slightly different arrangement. That's a better word to use. However, or slightly different position. That's a better word to use. But yeah, so we just drew several structures for nitrate. It's famous for its resonance, and let me just note something. When we're drawing different resonance structures, so say we have resonance structure A, resonance structure B, resonance structure C. There we go. So that's how you know you're drawing resonance structures, as opposed to, so the arrows, these are the arrows for resonance, and these are the arrows for equilibrium. I'm just knowing this because it's important to know the difference. So this is resonance. These are resonance arrows. When we're drawing resonance structures, these are the arrows we use when we're talking about equilibrium. So it's important to just note that difference. But I just want to show that to you all. But let's move on. Let's move on. Let me just erase, guys, and then we're going to proceed. Then we're going to proceed forward. Sorry about the background noise. Yeah, let me just erase. So it's important key ideas that I want you to take away from this exercise. You can draw different Lewis structures for molecules that tend to have double bonds and single bonds in the same structure. And it's important for you to know that the best structure or the best resonance structures you can draw minimizes formal charge, and it also follows the octet rule. That's the big thing. The big idea is make sure it obeys the octet rule, especially for second group elements. Second period elements, excuse me. Okay, so let's talk about. So this is just another thing I want to do is I want to make sure you understand how to study this type of material. Now, some people, when it comes to andragogy or learning when it comes to adults, some people may promote